Thank you very much. I'm honored, very honored to be a recipient of the Van Vlack Lectureship. And I'd like to start by describing my connection with Larry Van Vlack. And that is this textbook. And so Larry Van Vlack had this novel idea back in the very early stages of the development of our field that you could teach material science engineering to undergraduates at the earliest stages in their career. And that started really in the mid-'60s. By the time I rolled around, it was on the 4th edition. This is actually sitting on my bookshelf. And through all the moves over the years, you can see it's in reasonably good shape. But I think what I found very interesting about the book, and very prophetic actually, was the dedication. Is today's students who will be tomorrow's engineers. And I guess in this case, he was absolutely right. And so he not only influenced me tremendously, but also generations of students who have used his book. So I'm very grateful and honored to be here today. I have to start by thanking the people who actually do the work. The 3 graduate students in particular here. John Gibbs did the work on dendritic growth that I'll show. Aditya Mohan developed the numerical algorithms to do the tomography. And Ashwin Shahani, a current graduate student, did the other 2/3 of the work that I'll be discussing. And this is work in collaboration with a postdoc, Begum Golsoy, Charlie Bauman, who's in the electrical engineering department at Purdue, Mark DeGraff at Carnegie Mellon, and Zhenghui Zhao, who's at the beamline at Argonne National Laboratory, the Advanced Photon Source. So I want to talk about 3 subjects: dendritic growth and the growth of faceted crystals from a liquid, and eutectic growth. And I'll explain what all these things are as I go along. And I think everyone in Michigan and certainly in Chicago know what a dendrite looks like. This is a dendrite of ice growing from a vapor. It's commonly called a snowflake, but people like to be technical around here, so we'll call it a dendrite. And if you look at something like this, this is a solid that is growing from a vapor. The vapor is featureless. It has no orientation. Yet when the solid grows from the vapor, it develops this very, 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 very beautiful crystallographic, orientationally relevant shape. And so the question becomes is, why does it have this shape? If you look carefully at the branches up here, you can see that those don't follow the crystallography as well. In fact, they're relatively uncorrelated. And so why do the branches form? How do they form? And what controls the overall morphology? And so this is kind of the science justification for this work. The practical application is that most materials start their lives in a liquid. And you do casting to form a solid. And this is an example of some sand casting from that I took off the web. And what happens when you do sand casting is that you create, or any casting, is that you create dendrites. And this is a 3D reconstruction of a dendrite from our work in aluminum copper alloys done many years ago from serial sectioning. And this is what it looks like. And the dendrite basically forms from the liquid. It grows from the solid. It's present in all castings, and the interesting thing is there are about 1 trillion dendrites created yearly by the steel industry alone. That's a lot of dendrites. And what's important really is the spacing between these branches. And to put that in perspective, this is the width of a human hair on the same scale, so you can get an idea of the length scales here. And the spacing between these branches here, which is much less than a human hair, control the properties of castings. It's kind of an interesting thing that something so small can control the properties of something so big. And that's really the essence of material science engineering. And so what we're trying to understand is, since they have that strong link between the properties and the spacings of these dendrites, how do we control the spacing? What sets the spacing of the dendrite arms? And this is not a new question to us. It's been around for a long time. And people have been pushing at this in a number of ways. One of them is called quench and look experiments, where you take an alloy, you quench it, you section it up, and you can make a 3D reconstruction. And these branches here are colored by the local mean curvature, which I'll explain what that means in a minute. But the important thing to note here is that positive mean curvature is red and negative mean curvature is black. And we did it this way so it looks like a Christmas tree. The other way to look at these dendrites is to have them grow in situ and look at them through a light microscope. And this is a succinonitrile. It's a plastic organic material. The problem here is that you only see the projection. You only see the actual 2D projection. Here, you get the three-dimensional morphology, but you've quenched. And so the question asking is, how do you get around the problem that you can't measure the three-dimensional morphology in these experiments? Furthermore, it's not a metal, it's an organic material. And how do you actually avoid this quench problem? And the way to do that is to go to 4 dimensions. So we have to measure the evolution of a material in 3 dimensions and in time. And we want to combine these 2 experiments somehow. And one way to do that is by tomography. Many people have had the joy of being tomographized at a hospital. And the way it works is pretty simple. You sit here, an X-ray beam goes through you, and there's a detector on the other side. And this X-ray beam moves on this outer track around here, and the detector moves on the other direction, and it takes what I'll call 2D projections through you, and then reconstructs the interior of the body by making many of these 2D projections. The interesting thing here is that this thing rotates at relatively high speed and weighs about as much as a car. So it's a tremendous feat of engineering that it works as well as it does. We want to do this kind of experiment, but on something that doesn't transmit X-rays as easily as the human body in metals, and so we have to go to a machine that's a little bit more powerful. And this is called a synchrotron. This is the one at the Advanced Photon Source at Argonne. So this is a ring. To give you an idea of the scale, that little dot down there is a car. Okay? So this is big science. It's not something you do in the lab. And electrons whiz around this ring at almost essentially the speed of light, emit X-rays that come off the beam and that come off the synchrotron and are captured by these beam lines that are along the side here. And this is where we do the experiments. And so if you're doing an experiment in a synchrotron, you can't do what you do in a hospital. You can't rotate the advanced photon source. What you do instead is you rotate the sample. Okay? And so you have a furnace. This is our little aluminum copper sample, about 1 millimeter in diameter. The X-rays shine through, and you take a 2D projection of this image on the camera. And then instead of rotating the X-rays, you rotate the sample. And what you do is you collect these 2D projections at various times and then reconstruct the 3-dimensional image. The challenge here is that if this is evolving, each projection is a different structure. So imagine the challenge that doctors have when they're doing tomography of a heart that's constantly beating. So each one's a different projection. So this can make the reconstructions very difficult. And to get around that problem, to address that problem, we developed this new algorithm that's a time-interlaced model-based iterative reconstructions. And I won't tell you the details of the mathematics, but if you're interested, it's in that paper there. So it's an iterative way of basically doing reconstructions in 4 dimensions, where we take into account all the data that we collect on the camera. And I'd be happy to talk about the details of how the algorithm works later. But it's actually essential to the experiment. So here's the experiment. So we start with a liquid up here, solid over here, solid down here. This is a solid liquid region in here. This is sometimes called a mushy zone, where you have mixtures of solid and liquid. You want to think about this as a slush, this is a liquid, and that's the snow. So you cool down from high temperature, and we're going to watch the dendrites form as you get below this line of the liquidus temperature, and you're in this 2-phase field. So you cool the sample from a liquid relatively slowly, so everything is essentially at uniform temperature. 1 millimeter diameter samples, voxel sizes. Okay, so everyone's familiar with the camera. If you go to a photography store and you buy a camera, they're measured in pixels, the number of pixels. Since we're doing things in 3 dimensions, we talk about voxels instead of pixels, and they're 0.75 micron voxels, to give you an idea of the resolution, and the data sets are on the order of 1024 cubed or larger. The challenge for us is that we have the true big data problem. So a typical experiment will generate on the order of 14 terabytes worth of data. And this is sufficiently big that we can't transfer this data over the internet from the APS to our lab in Chicago. And so what we do is that we show up to the APS with many portable hard drives. We put the hard drives in the back seat of my graduate student's car. He drives it to Evanston, and we get a faster transfer rate than the network. So you never want to underestimate the baud rate of a graduate student with terabyte drives in the back seat of a car. Okay, so we have to figure out how to transfer this data, handle this data, share this data, and that's a real issue for the community that's coming. So this is our 1 millimeter diameter sample. This is liquid, and that's the growth of an aluminum dendrite from the liquid. It doesn't look very much like snow, and I'll explain why in a second. We'll get there. But you can see it's a highly complicated structure. It grows from one side to the end and eventually crashes into this end of the sample when the supercooling is expended or the supersaturation is expended and begins to slow down. So we have this time resolved about every 1.2 seconds in between each one of those frames. And so we can go back and pick out one of the frames, and this is what the dendrite looked like at that particular time. And so it nucleated at the edge of the sample over here. That's where it nucleated. And you can see it still has some evidence of crystallography. This is a cubic structure, a material with a cubic crystal structure, so it has fourfold symmetry to it. And you can see that there's a trunk here and the side branches come off the sides. And so given that we have this structure, we can begin to analyze it. And we can begin to quantify what it looks like. So this is an example of another microstructure, but it's here to prove a purpose. This is not what the computer sees. What the computer sees is actually this. And the way computers do renderings is that it breaks up the surface of a structure into small little patches, and it does a light tracing algorithm that puts shadows into the pictures. And so what we use, we're not so much interested in how the ray tracing works. What we're interested in is the individual shapes of these patches, because that tells us something about the overall shape of the structure. And so you want to think about what I'm going to show you next is we go into this structure, we have these little patches, and we just take each one of these patches out of the structure and we ask, what is the shape? Does it look like a patch of a sphere? Does it look like a saddle? Does it look like a cylinder? And we can begin to quantify the shape. So these are patches of various shapes, and we have a parameter that takes us from something that looks like a sphere to something that looks like a cylinder, to something that looks like a saddle, literally, with the legs of the saddle here and the backs of the saddle here, to cylinders and spheres in the opposite direction with respect to the liquid. The other thing that we've defined is a curvature that is basically the average of the principal curvatures of the surface. This is very easy to think about. This is a needle. The tip of the needle has a very high curvature, so C is very high. A desk, like this, has zero curvature. Okay? So we can map everything from a zero curvature shape to a needle in terms of the overall curvature, and then the shape sits here. And so we can go into this dendritic structure, pick out all of these patches, and plot the probability of finding a patch of a given shape and a given curvature relative to the average. And I won't dwell much on the details here, but suffice it to say that if I look at the shape of the dendrite up here, this is what the curvature distribution looks like. So there are large numbers of patches that are cylindrical in shape, which are the edges of the dendrite arms. And if I go farther back, it looks somewhat similar, but it's really quite different. There's a much bigger intensity here, which is associated with the development of the dendrite arms that I'll show you, and also large numbers of patches down here with near zero curvature. And I'll show you why that is in a second. Okay. So if you look back at the secondary arms, so this is the trunk. This is 25 microns to give you the resolution, idea of the resolution. This is the trunk. These are the secondary arms, and they look kind of like American pancakes, not French pancakes, right? So they're a little thick along the edges, but very flat on the tops and bottoms of the shapes. And so if you look at this distribution function I showed you before, you had large numbers of shapes with near zero curvature, and those are the tops of these dendrite arms. And the edges along here that look like cylinders are these patches that sit along here. So we can quantify the shapes, and we can basically use this as a test for theories and simulations of dendritic growth. The interesting thing, however, is if you compare these shapes to this, they don't look very much alike. In fact, the interesting finding of this is the metallic dendrites grow in a way that are very different, or that are different than succinonitrile dendrites that have always been kind of the standard reference material for dendritic growth experiments to date. And it's because we can actually see what's going on in a metal that allows this. So these secondary arms grow like in a pancake, and the edges of the pancake go unstable. And what's interesting is that it develops these large indentations along here. And if you let it grow longer, you actually end up with something that's called a doublon in the community, where you have a double-shaped dendrite tip with this very long reentrant groove here. And this very long reentrant groove actually traps solute. Remember I said that the properties of a material are related to the spacing? Well, the spacing here is much, much different than the spacing here. And so you get a different distribution of the elements in the alloy in here than you would out here. And this, again, affects the properties. And so the message is that the secondary dendrite arm spacing, or this spacing here, maybe not is what's better correlated with the experiments, but you have to look for situations, with the properties, but you have to look for situations like this. And this is very common. If you go down the dendrite stem, you have doublons and doublons and doublons. It's actually occurring quite often. And the answer is why? We don't know. We don't know. This is still an open question in the community. We don't really know why they form these doublon structures. Okay, so let's go back to the ice and the aluminum. And so if you look at these, it's pretty clear that they're very different animals, right? This one has a fourfold-like shape. It's three-dimensional. This thing, as you remember what a snowflake looks like, is truly two-dimensional. This is really a disk. So if I took this and I rotated it this way, it would be a very thin disk. It's really two-dimensional. It's really flat. And it only develops these arms in a particular plane, whereas this one seems to develop the arms in a more of a three-dimensional fashion. And so why is that? Well, that's something we actually do understand. If you look at the solid-liquid interface here, so these are the atoms along a solid-liquid interface. This is a molecular dynamics simulation. So this is the crystal over here, and you can see that they're all lying in a very crystallographic nature. They're all lined up in great order. The liquid is highly disordered. And this interface here is what is called atomically rough. It's very easy for an atom in the liquid to join the solid because it's atomically rough. It can find places to stick. If you look at this structure, it's actually what's called a facet, and this is the crystal structure of this of ice. It's hexagonal. This is the face. The basal face here is what's actually on the face here, and it's very hard for atoms to attach to the face of this crystal because it's atomistically flat. Whereas on the sides, it's atomistically rough, and it can grow these nice secondary arms. And so if you blew up what the basal face looks like here and looked at the atom structure, it looks like this. It's completely flat. And so you want to think about this like a skating rink, an atomic skating rink. Atoms come down, and here they really can't stick. They just kind of slide right off the side. It's very hard for atoms coming down from the liquid to actually form the next layer of solid, whereas over here, it's relatively easy for the atoms to join to the solid. So this makes it hard for the snowflake to grow perpendicular to the branches. It's this faceted nature of the crystal growth process. And so how do facets move? They have to form a layer of atoms somehow. And the simple way is, well, you just rain the atoms down from the top, and they eventually form a small little pillbox nucleus of solid, and then the atoms grow, and the atoms attach to the ends of the steps, and the steps go across and you form a facet. Turns out that that's very difficult to do. It requires very, very strong driving forces. What usually happens is that you have steps on the surface, the atoms come down, and they migrate along the surface, the terraces, and they attach at the edges, and then these edges propagate along. And so basically, if you have steps on a facet, the facets can grow. And that kind of begs the question, right? Where do the steps come from? If this is difficult, then if you don't have some mechanism to create steps, the facet doesn't move. And that's almost what happens in the ice case. And we'll see what happens in silicon. So another important alloy system is aluminum silicon, or aluminum silicon copper. It's 90% of all casting alloys. It's used in engine blocks and all sorts of other things. It has beautiful properties. And if you cut the engine block open, it looks something like this, and look at it in 2 dimensions. You have these silicon particles here, silicon plate-like particles in a matrix, and I'll talk about that in a second. But what's clear is that these silicon particles are highly faceted. Now, this one looks like the edge of an X-Acto knife, for example. Very, very flat face and very, very sharp points. And so these are growing as faceted crystals. And the question is well, okay, these are faceted. We know that in ice, if it's faceted, it doesn't grow perpendicular to the basal plane. But in this case, the silicon is growing. So how does the silicon grow, and where do the steps come from? And the steps come from, in this case, twins. And so if you take a crystal lattice here and create a twin defect along here, and it really is a twin in the truest sense of the word, that if you put a mirror along here, this side of the crystal looks exactly like this. So it is a mirror twin. And in 3 dimensions, it looks something like this, where the atoms go up and down and up and down. The atomic planes go up and down with the twin defects here. It turns out these twins are very important in the growth of silicon, as we'll show you in a second. And because if you go back to the 1960s now, this is all the way back to the 1960s in the Bell Labs days, people speculated that the way silicon grows is by the presence of these twins in the silicon plates. And so you have one twin here and one twin here, and you form a little nucleus here easily because it's associated with the twin. And this nucleus propagates along, and then you found another twin here, and it again propagates to form the new layer. And so the key idea in this mechanism is that you need 2 twins, and that the twins are the defects that provide the steps to allow this crystal to grow. Okay? So there's a strong correlation here between defects and crystal growth. In this case, it turns out to be twins. But there are other theories. The great thing about science is there's no data. You're going to find a lot of theories. And so there are lots of theories on how this might happen. There's one by Fujiwara. This is in the year 2000s. Lee and co-workers have yet another theory for how silicon might grow. And basically the problem is you can't see the structure grow in situ. Everything's being done by analysis of the structure after the fact. So we go back to 4 dimensions. So this is aluminum, copper, silicon liquid. This is the crucible here. There's a time counter here to give you an idea of the time scale. And the primary silicon phase nucleates at the edges of the crucible and grows into the liquid. And you can see it grows in. They have a truly faceted, very plate-like structure here. If you look down here, it doesn't look so faceted, right? So the question is, why is that true? And I'll talk about that in a second. So basically, we can capture the growth of these individual plates and begin to say something about how the plates grow into this aluminum silicon alloy. And we're going to focus on this region over here. So that region of the structure. And so we focus now on just a small little area. And so the silicon plate grows up, hits a point, and then begins to grow up faster. Okay, so I'll let this run a couple of times here. So the first thing you'll note is that this doesn't look like a snowball, right? It just doesn't grow up isotropically. It grows up highly anisotropically, and it's basically bounded by 2 facets. That's this one and this one that are 111 facets for the materials people in the audience. And these things have very slow growth rates. This is just like the basal plane of snow. It does not grow very fast. But look at the edges. And the reason why this grows so fast is if you look at the edges that are bounded by these 111 planes, it grows much, much, much faster. And we're going to take a look at what's going on in this region here. So since we have time-dependent data, we can measure the interfacial velocity. So we have low interfacial velocity over here, high interfacial velocity over here. And I'm going to direct your attention right to the very edge of the sample over here. So the edges here, so this 111 plane's growing slowly, that 111 plane's growing slowly. The edges here are growing much faster, and the question is why? Why are the edges growing faster than the 2 sides? Well, if we know that this is a slow-growing 1-1-1 facet, and the other side is a slow-growing 1-1-1 facet, since we have the shapes, we can begin to index the orientation of each one of these facets. And this is what, in fact, Ashwin did. And so this is the stereographic projection for the materials people in the audience. But the key idea here is that the orientation of the interface is a function of the colors that you'll see on the reconstruction over here. So now I play the same thing, but now I'm playing, give you an idea what the orientations of the surfaces are. And so the surfaces here are blue. That's the 1-1-1, as you'd expect. And if you look at the edges here, you can see the stripes that are forming near the edges of the crystal. Let me run that again. So the crystal grows up. You have the slow-growing 1-1-1 planes, and you form slow-growing 1-1-1 planes. These are the basal planes that don't grow very fast. And then these planes, these two lines come together and split into these lines here. If you look at the crystallography of the orientation, in other words, the orientation of the interfaces, you see this. So what we did here is that we took the trace of the solid-liquid interface here, and we looked at where those twins, where those boundaries actually intersected the solid-liquid interface. And so we put a dot every time we saw where it intersected the solid-liquid interface. So this is a function of time going along here. And you can see that these dots lie in a relatively straight planes. And you can see that the interface is kind of non-planar as it crosses these dots. And these dots are actually twin boundaries that we can get from the experiments. And so if we go back and we connect the dots, so to speak, you find these planar boundaries, and these are the twins. So, and the solid-liquid interface have angles with respect to the twins that are exactly what you'd expect. So these are 1-1-1 planes. And the twin boundaries are intersecting here. So this is the picture I showed you before. And so this is actually going into the crystal in this direction. And these are the twins that the projections of which are in the plane of this crystal. So we've rotated around. So in this case, we're looking down on the crystal from over here. So you can see the extent of the twins. And what this means, really, is that this is confirmation of the mechanism that goes back to the 60s, where what happens is that these 1-1-1 planes are crystallographically perfect. So remember the picture I showed you before with the ice. It's essentially like a skating rink. It's impossible for the atoms to attach. So what happens is that these are very, very slow-moving interfaces, and they would never move, except for the fact that we have a twin that intersects the surface, and this twin acts as a nucleation site for the next layer of atoms. And it's very easy for the atoms to form at the twin, and it propagates along in exactly the mechanism that Seidensdecker postulated back in the 60s. And so we were able to confirm this growth mechanism in this aluminum silicon copper alloy that had been out in the literature since the 60s. Now I will say that this is a very particular experiment in the sense that the temperatures are relatively low. So it's possible that these other mechanisms that have been proposed are in fact correct, but they're at different temperature regimes or higher temperatures where the nucleation characteristics of steps on twin boundaries might be, for example, a little different. Okay, so let's go back and look at these aluminum casting alloys again. So as I said, you have this very nice mechanical properties. And if you look at the structure in here, you notice that there's some, it's not a uniform color in the etch after it's etched. And if you blow up that structure, it actually consists not of a single phase, but a mixture of aluminum and silicon. And it's called a eutectic in this case. So it's aluminum, and actually the copper goes in mostly entirely into the aluminum phase. So it's basically aluminum and silicon. And so you want to think of this structure here as something that's somehow intermixed. And so how does aluminum and silicon become intermixed to the extent that you see the microstructure here? How how does it mix, how do these two phases mix together? And it does this by a cooperative growth mechanism. And so again, we go back to transparent materials that make it easier to see. And so this is a liquid, this is a solid, and it turns out to be carbon tetrabromide and some other chemical compound that I can't pronounce. I'm sorry, I'm not a chemist. And so you're growing this from the liquid, and so this is one phase here, and this is the other phase here. And the reason why this grows cooperatively is that the solubility of these two components in each one of these two phases is different. And so in one case, it's rejecting the material, and it's going over to here, and it's going over to here. And so it needs these two phases to be intimately connected with each other such that the rejection from one phase is sucked up by the other. And you get this beautifully regular structure, except for this one little defect there, but you get a beautifully regular structure. And this is what happens when you have a nice isotropic system with no facets. That's clearly not the case in silicon, right? We already established that they're facets. And this is from some beautiful work that was done in France by Silver Akamatsu and Gabriel Favre back in the 1990s, late 90s. The other problem with these experiments, the other thing you should understand about these experiments is that they're done in a thin sample. And so what you're looking at here is you're looking down, actually through a light microscope, at a thin microscope slide. So if I took this out, the microscope slide would be very thin. And here's a schematic of this. You're looking down on top. Here's the solid. Here's the liquid. This dimension here is about 10 microns. It's a very, very thin cell. Okay? So you're constraining the growth process to be within this cell, because if you do things in 3 dimensions in a transparent system, you can't see anything. It's just impossible to understand what's going on. And so we have this problem that we faced in the dendritic growth experiment as well, that you have these beautiful analogs, but we have to look at these again in 2 dimensions. If you go to metallic samples in faceted structures, this is aluminum silicon. You quench, and this is the eutectic here, and this is what used to be liquid. And the whole question is, well, what are the effects of the quench? You don't know. This from Angus Hallowell in the 70s. People tried to do faceted organics so that you can actually see and not avoid the eutectic, and this is what you get. This is from some classic work by Wilfried Kurtz in succinonitrile borneol. And the question here is, does this look anything like what you see in the important thing that we're really interested in, which is aluminum, silicon, copper? You need to go to 4D again. So you need 4-dimensional experiments. And so here's the growth of the eutectic. We didn't do aluminum silicon, we did aluminum germanium. It behaves exactly the same way. The aluminum phase is white, the germanium phase is green. And you can see that again, it's cooperative growth. You cool the liquid down, and you'll notice that you can even see at this very low resolution that the germanium and the aluminum grow essentially simultaneously. They grow together. They're coupled together. So you have this coupled growth. And now that we have this data, we can begin to look at what's really going on during the growth process. So we're going to take one small sample of the section over here and look at that in detail. And this is a eutectic colony. So this is again germanium. This is aluminum. The reason why we're using aluminum and germanium and not aluminum silicon is that it's very easy to do the tomography in aluminum germanium. It's very hard to do the tomography in aluminum silicon because the atomic numbers are so close. But for all intents and purposes, they behave essentially the same, aluminum and silicon. So this is the front face, and this is the back face. This is the side. So this is the front, and you turn this 90 degrees, and you can look on the side here. And so we can now turn this on. So it grows up, and in a few, this is about 100 seconds, occupies the whole frame. So the first kind of takeaway general message here is, again, it's not growing like a snowball. So if you nucleated an isotropic growing solid in a liquid, it just kind of would grow up like a spherical object. Here it's not growing like a spherical object. It's highly anisotropic. This plane here is a 1-1-1 slow growth plane of germanium, just like the 1-1-1 slow growth plane of silicon is doing precisely the same thing. It's very hard to create steps on the surfaces of the germanium, so it grows very slowly. And therefore, what happens is that you have this very highly anisotropic growth process where the width doesn't change very much, but it grows very fast in the other direction along the rim here. So this is aluminum. The white's aluminum, and the orange is germanium. So let's look at a little more details to what's going on here. Okay, so I'm going to step through the frames. The first thing you'll notice is that this is a germanium plate, and the aluminum is kind of growing almost like fingers along this germanium plate. So it's very, very, very much unlike that beautiful picture I showed you before of the organics, where everything is very oriented and growing in a very, very highly periodic fashion. What I want to direct your attention to is look at the edge of this plate. It doesn't look very plate-like at the edge, and that's kind of important. And in particular, watch what happens here. Watch what happens in this region. So there's a little bit of aluminum here. The next time step, that's grown bigger. It stopped the growth of the germanium plate, and then that aluminum began to grow across the face. And so you have a cooperative growth process, but where the aluminum is actually working its way through holes in the germanium plates. And so that's one example over here. There are other examples up here. We watch these things appear through the plates. And so there was a large controversy in the community back in the 70s, where people said that the way this eutectic grows is that it nucleates aluminum islands independently of the growth of the eutectic, and it's not cooperative growth at all. And the answer is that is not the case, but it is polycrystalline because these aluminum islands come from various parts of the sample. The other thing is, if you look at the side view, these are the germanium plates, and here's the aluminum. You can see where the aluminum goes over the edges of the germanium plates. And I mentioned at the beginning here that these germanium plates are not very flat. They're highly rough. And the reason we know that is true, that's not an artifact of the tomography, is because they're actually, these germanium plates are highly defective. They're full of twins. And the way we can see that is that this is one of the germanium plates that go, here's one germanium plate, here's another germanium plate, and then you can see on the sides here other plates that are oriented at exactly the angles you would expect if they formed by a twin formation mechanism at this point. So what happens here is you have a twin that forms, and this twin then allows another germanium plate to grow out at an angle. And so that's how this plate actually extends its size. It doesn't extend laterally by actually growing the germanium. It actually extends laterally by forming these fin-like structures in the material. And this is consistent with what Angus Hallowell conjectured on the basis of metallography a way back, and inconsistent with what a group in Australia has been saying for many years about how this structure grows. Okay, so what happens is that you have a germanium plate here, the aluminum phase, the aluminum metal grows up, and it goes through the hole and begins to propagate along the outside. And so you have cooperative growth, but it's nowhere near what you would see in the case where the interface is not faceted. And so if we go back and we look at what people have seen in the literature, this is what people would see on the basis of a quench. So you take a eutectic that's growing and you quench it, and you quench in the liquid, and this doesn't look anything like this. This is what you see in the organic that was supposedly an analog of this. You see some remnants of similarity in the sense that you have these faceted like needles here, but these don't look like needles, these are plates. And so what happened in these experiments is, again, as I showed you, that these were done in a thin 2D sample. very thin plates. And the faceting direction was not well aligned with respect to the growth direction, and they were constrained by the plates such that you couldn't see the development of the structure like this. And so you really needed three-dimensional views of the structure in order to be able to understand how these things grow. And both the quenching experiments and the organic analogs did not provide a very good view of the structure. So what have I shown you? So I've shown you that in situ tomography can in fact provide information on the evolution of microstructure and technologically important materials. I've talked a little bit about dendrites and the insights we can get into the dendrite morphology and the insights and basically the benchmark data sets we can now use to compare simulations of dendritic growth to experiment. We've identified a mechanism for the way silicon plates grow that is a confirmation of the twin-enhanced growth mechanism that goes back to basically the reentient twin growth model of Seidensdecker. And in the germanium aluminum eutectic, we've shown that these faceted eutectics grow in ways that are really quite surprising and had never been seen before. But it's still in a somewhat coupled fashion where the aluminum and the germanium are still following each other in some sort of very, very approximate sense. So thank you very much. It's been a pleasure to be here, and I'd be happy to answer questions. Thank you.
It's kind of like how the computer and electronics industry advanced. In the old days, yes, you could do computing, but it was a big room full.